For those of you who have already arrived, we're just going to wait a couple minutes for folks to make their way into the webinar. All right, well, let's get things started here, and the last few people will probably be trickling in as we get going. So on behalf of the BioMaker Space and the BioMaker Student Group, I'd like to welcome you to the next speaker in our series, COVID-19 BioMaking Solutions seminar series. A couple quick notes before we begin. Want to just note that we've got one more talk lined up for next week. Dr. Patrick Boyle of Ginkgo Bioworks, so make sure you register for that talk and attend there as well. Up on the slide as well, there is contact information if you have any questions about the BioMaker Space or the BioMakers Group, as well as links to the website for the series where you can register and see previously recorded talks. As far as format for today, please hold all the questions to the end. When you have a question, please use the raise hand button. I will then be able to bring you off mute. You can ask your question directly to our speaker. And if you have any problems with that, just let me know and I will try to assist you. So with that, I'd like to hand things over to Professor Belcher to introduce our speaker.
Well, thank you. I'm excited to welcome everyone to the BioMaker Space today. So welcome to, oops, my picture's not up there. I'm excited to welcome everyone to the BioMaker Space today, and welcome to all those biomakers out there. I think that today is going to be another exciting talk, very timely talk on from Professor David Gifford from MIT. He is a professor in electrical engineering and computer science and the Department of Biological Engineering. So the interface between these two field is becoming even more important, and I think even more important in the face of diseases like COVID, which we'll hear about today. David received his bachelor's degree here at MIT in computer science and went on to earn his master's and PhD in computer science and electrical engineering at Stanford. He's a member of many has many other affiliations, including the Broad Institute and HST. And he has affiliation, as I said, in biological engineering. So today he's going to talk about some pretty exciting work in the interface between machine learning and therapeutics. So I'm going to turn it over to David.
Well, thanks a lot, Angie. I really appreciate that. And let me share my screen here so people can see. I presume that's working okay. Everybody can see that?
Looks good.
Oh, great. And so this is a joint work between myself and members of my lab, Guy Liu, Brandon Carter, Trenton Bricklin, and Sid Jane, as well as some folks from the Ragon Institute and the NIH, Mary Carrington and Matthias Viard, which this work has been recently published in part in Cell Systems. And just to provide some context about COVID-19, I thought it would be useful to share some recent statistics about the fatality rate of this disease as of yesterday. And of course, this could be overestimated given that certain people present asymptomatically with the disease. But as you can see, in the United States, about 3.3% of the cases are fatal, whereas in the United Kingdom, surprisingly, the mortality rate is much higher. This just reemphasizes how important addressing this disease is as sort of a global pandemic. Today, I'm going to address 4 questions about COVID-19. The first question is, what fraction of the population do we need to protect to actually be able to go about our business as normal, more or less? How are COVID-19 subunit vaccines designed? How can we improve these subunit vaccines? And how can we make COVID-19 peptide vaccines that are subunit free? So let's turn to the first question of what fraction of the population do we need to protect? So a way to think about this is in terms of the R0, which is if we take a given person, how many other people they're going to infect on average. Of course, this is influenced heavily by social distancing, but let's just take the case where we're not socially distanced and ask how many people on average different diseases actually typically, how many of the people we would infect for a typical disease like Ebola. It's 2. For SARS, it's about 4. COVID-19 is actually even more contagious than SARS, so its R0 is probably around 5. And a way to think about what fraction of the population needs to be immune can be visualized as follows. If we assume an R0 of 4, each person would go on to infect 4 other people, and each one of those people would infect an additional 4 people, leading to exponential growth. However, if 75% of the population is immune, then each person would go on to infect 1 person, and that person would then go on to infect 1 person, and so forth, and thus exponential growth of the disease would be stopped. And thus, approximately 1 minus 1 over R0 of the population needs to be immune to stop exponential spread. So that would imply for our estimates for COVID-19 that we need to have, roughly speaking, about 70 to 80% of the population immune to be able to continue our life without social distancing. Let's turn to the next question then, which is how are COVID-19 subunit vaccines designed? In order to approach this, let's just begin with some review of immunology and how COVID-19 is treated by our immune system. Because what we would like is for a vaccination to, in essence, replicate the immune response that we would see from the COVID-19 virus itself. There are 2 kinds of immune responses that we are concerned with. The first you've heard most about is an antibody response or humoral response, where a B cell receptor that recognizes the viral particle activates B cells, causes them to expand, secrete more antibodies, and to induce B cell memory. And this is assisted with T helper cells. The other branch of the immune system that we are concerned with is the cellular immune response, where infected cells display on their surface bits of their protein garbage can on MHC molecules, here MHC class 1. And our CD8 positive T cells are trained to recognize these non-self molecules and ultimately lead to their destruction. So one way to think about this is that the antibody system is dealing with these soluble viral particles and actually neutralizing them, whereas our CD8 positive T cells are patrolling our bodies and killing cells that are infected. And thus both kinds of immunity are important, the antibody-based immunity and the cellular-based immunity. Note that the T helper cells require presentation of these viral peptides on MHC class II molecules. So to get the appropriate assistance of the CD8 positive cells and the CD4 positive cells, we need to have our viral peptides displayed on MHC class I and class II molecules. And one nice property of the CD8 cells is they can actually provide sterilizing immunity. That means that as they patrol our bodies, they can recognize infected cells, and the ones that actually are displaying these viral peptides can be killed by the cytotoxic T cells. So this kind of response is important for durable immunity. Okay, so how can we induce this kind of immune response from our system without being infected with COVID-19? Well, here's the SARS-CoV-2 virus that causes COVID-19, and we can select a subunit of this virus and use that subunit to induce an immune response. Now, if we examine the functionality of SARS-CoV-2 and how it enters our cells, we find that the spike glycoprotein contains a domain called the receptor binding domain that binds to the ACE2 molecule on our lung cells and uses that to jump inside of our cells, in essence. And so if we look at the way this has been modeled, you can see the spike protein interacting with the ACE2 protein, also shown here right in this panel, and thus neutralizing antibodies that bind to the spike protein at the RBD domain can disrupt the ability of the virus to actually infect our cells, thus getting neutralizing antibodies that interfere with this process is part of the immune response we really want to see from any kind of immunogen that we select. Now, vaccines can be characterized both by their payload, that is what immunogen they're delivering, and their delivery vehicle. And it's important to separate those two things, right? We hear a lot about RNA vaccines, DNA vaccines, and so forth. Those are all delivery vehicles. What we also are concerned with is the payload, what immunogen they're delivering to basically train our immune system to recognize aspects of the virus. So later on, if we're exposed to the virus, our immune system is primed to be able to deal with it effectively. Now, one consideration for choosing an immunogen is we would like to focus the antibody response on the part of the protein that will block its entry into a cell. We don't really need to have antibody responses to other parts of the viral component that actually are not going to be neutralizing. They would be less effective. As a consequence of this, we will pick immunogens that are going to produce these neutralizing antibodies. But that said, the COVID-19 vaccine problem is challenging. Vaccine design has failed for a number of different diseases, and as you're undoubtedly aware, it's not completely effective for flu, even when we actually match the strain of flu that circulates exactly. Another challenge is that picking model systems to mirror human presentation of the disease is also a problem because these model systems don't completely replicate our immune system, and in particular the MHC molecules that are contained within it. In addition, trials with the original SARS vaccine that were a consequence of the 2003 outbreak indicated that they really don't completely protect aged animals. They were aged 1 year. And furthermore, sometimes when you get vaccinated, what happens is that when you are later exposed to a virus, it actually causes a separate kind of disease. And in these models of SARS, there was a immunopathology associated with lung necrosis when the mice were vaccinated and then were subsequently exposed to SARS. And finally, recent animal studies from rhesus macaque show that spike protein vaccines don't eliminate virus from nose swabs in these model systems. So as you can see, the vaccine problem is quite challenging. Now, if you look in some of the most recent clinical trial data and you dig back in the appendices of these studies that have been published, for example, this one that came out about 2 weeks ago, you find some interesting data. This is the only study that has presented age group segregated efficacy of a vaccine here based upon the S subunit. So they vaccinated individuals with the S subunit and asked what fraction of the folks seroconverted to produce neutralizing antibodies that could stop SARS-CoV-2 from infecting cells in vitro. And what you can see is that while the seroconversion rates for younger groups was quite effective, for people over 55 years old, the seroconversion rates were quite low, which of course is a concern and asks how could we help boost the efficacy of an S subunit vaccine. Now, if you look at all of the warp speed candidates being funded by the US government, you can see that all of the reported ones use spike protein subunits, and Vaxart has not reported what their particular formulation is. And thus, there is a fair amount of risk in some sense concentrated on using this single subunit as a vaccine immunogen in all of these constructs. So, how are COVID-19 subunit vaccines designed? The answer is that right now there's an S subunit design commonality across all these reported warp speed candidate vaccine designs. So, how can we improve these subunit vaccines? Well, let's turn to how we could actually amplify cellular immunity when a person is presented with a COVID-19 vaccine. To do so, let's think about the presentation of vaccine peptides by the human MHC system. Here we're going to talk about the presentation on class 1 molecules. Here the classical ones, HLA-A, HLA-B, and HLA-C. You get 3 of these HLA molecule alleles from your mom and 3 of them from your dad. And there are thousands of different alleles, and each allele has a particular preference for the peptides that it's going to display from the virus. So if we think about this, we can use machine learning methodologies using a great deal of training data that's been developed over the past 20 years to predict for all of these alleles which of the vaccine peptides or peptides drawn from the virus they're going to be displaying. And so, for example, here's one peptide and another one that's being displayed by HLA-A0201, and a disparate display profile is presented by this HLA-C02 allele. So given these predictions, we can parse the viral proteome into discrete peptides and then make predictions about what's going to be presented at a particular affinity threshold. Here, the threshold we're using is 50 nanomolar or better binding. It's important to recognize, however, that just because a peptide is displayed does not mean it's necessarily going to be immunogenic. And thus, we need to consider the fact that we may need to display multiple peptides by an HLA molecule for our cellular immune response to be able to recognize it, given the particular distribution of T cell receptors that we have. Now, going back to our diagram of the class 1 HLA system, you can see that of the peptides from the virus, we have 6 different display opportunities here, or we call these peptide-HLA hits, which is the number of unique peptide-HLA binding events in an individual that we observe. And so given a particular genotype, which is the combination of two haplotypes, we can predict the number of hits that we're going to get from a candidate set of peptides. Now, one unique aspect of our work is that we worked with the NIH to actually determine the haplotype frequencies of different HLA alleles. Now, because they're located close to one another in the genome, they're inherited together, and thus considering the marginal probabilities is not the best way to model this. So, for example, there are approximately 1,200 different class 1 haplotypes in a population that self-reports as black. And you can see here the haplotype frequencies for these different combinations of alleles at the class 1 locus. So we take these haplotype frequencies and we compute diploid frequencies of the appearance of these different HLA alleles. So obviously there are over 1 million different combinations for an individual self-reporting as black. And we can compute the probability of each particular genotype given the haplotype occurrence frequencies. And thus, for each particular combination of haplotypes, we can compute the number of peptide hits that we observe. So over the entire population of individuals, we can compute the expected number of hits that we will see in an individual. So computing this over all genotype frequencies is obviously a computationally intensive task that requires a few tricks to be able to do it efficiently. And if we look at the entire virus and ask how many peptide MHC hits does it produce if you had an infection of COVID-19, we would expect and predict that it would produce about 366 MHC class 1 hits and over 3,000 class 2 hits that would be observed by our immune system. This is in a non-redundant sampling of the COVID-19 proteome. That means that we're going to take windows that may overlap. So this can be an overestimate of the number of peptides that are being presented. But it's a point of comparison. And now we ask, if you take a subpart of the virus, a subunit, which of course is going to exclude a lot of viral peptides, how much does that decrease the footprint that the vaccine is going to present to the immune system? So we did that analysis for all the possible subunits of SARS-CoV-2. Here are 2 of them. And what you can see is that if the x-axis is the number of required hits, the y-axis is the uncovered fraction of the population that does not display that number of hits. What we can observe is that as the number of hits increases that are required, the fraction of the population that does not display that obviously increases. S subunit includes the RBD subunit, so it actually has a larger footprint to the immune system. But still, there is a sizable fraction of the individuals that don't display, say, over 5 hits for MHC class 2 for the S subunit. And it's even more pronounced over here for RBD alone. Now, the exact relationship between how many hits are presented in an individual and how effective a vaccine is yet to be determined. However, if you have a small number of, say, MHC class 2 hits presented in an individual, for example, 0, that would tend to suggest that the immune system is not going to be able to see the virus when it actually is presented. So how can we fix this? How can we improve the immune footprint of a vaccine to improve the presentation of pathogen peptides on MHC class 1 and class 2 molecules? Well, what we can do is we can take our starting set as the set of subunit peptides that's going to be presented as part of the subunit vaccine and ask, can we select other peptides that are not in this subunit to create a compact set of augmentation peptides that are going to improve at vaccination time the presentation of SARS-CoV-2 peptides that will then reappear if we're ever exposed to the virus itself. And we want to do this in a compact way. So we can perform this design task using a problem we call maximum n times coverage, which is a new class of problem in combinatorial optimization that we introduced to look at this task. So if we're provided with a set of HLA genotypes and we have a weight for each one of these genotypes, which is a population frequency, and we have a set of peptides that overlays these genotypes a certain number of times, how can we pick which peptides or overlays to use to optimally cover all of the genotypes and get a very compact set of peptides that are going to be effectively displayed, or at least predicted to be displayed, at vaccination time? So a way to think about this is to solve this combinatorial coverage problem. We can think about genotypes and their weights as their fraction in the population, and the peptide overlays as the number of peptide HLA hits each one will produce in each one of these particular genotypes. So for example, this peptide will produce 3 hits in genotype X1 and 1 hit in genotype X2, and A2 will produce 2 hits in each of X3 and X4, and peptide A3 will produce 3 hits in X2 and 1 hit in X4. We can then select which peptides to use and compute the number of peptide hits we're going to get on average across the population. And as you can see in this particular case, if we select all 3 peptides, we're going to get much better coverage. It turns out that this task is something that is a technical term. It's not submodular, which means that if we design an optimal set of peptides, say, that consists of 10 peptides, and then we ask, can we design another set that has 11 peptides, you might think that the 11th would simply be added on to the 10 set, but that's not the case. It turns out that if we add another peptide, that the entire set can change. So using this combinatorial optimization strategy, which as far as we can tell is a novel approach to doing vaccine design, we can pick optimal vaccines in the context of a fuller pipeline. And our pipeline considers, in this particular case, 25 MERS as peptides that we're going to present as part of the vaccine. We identify within all those 25s any self-peptides and eliminate those to avoid any kind of introduction of autoimmune response. We parse out all of the possible class 1 and class 2 peptides for consideration. We have sampled 4,690 different SARS-CoV-2 genomes worldwide, and that we can tune the mutation rate that we allow. Now here we're assuming that a low mutation rate indicates it's less likely that this peptide is going to drift over time. It may be necessary for function, and thus we're hopeful that that will produce a vaccine that will be and continue to be effective. And we can eliminate peptides that are glycosylated, that contain cleavage sites, or that are contained in parts of the proteome that we don't want to consider. We then can score all the peptides for their binding to all the various HLA alleles, and then we can perform our joint optimization here to maximize population coverage given a target number of peptide HLA hits, and then later on evaluate our vaccine using various metrics. So with this approach, we can add augmentation peptides, and here's a plot showing the number of additional 25 MERS that we add to the spike subunit and how it decreases the uncovered frequency in the population. And what you can see here, the different lines correspond to MHC1, MHC2. MHC2 are the dotted lines, and the color indicates the number of required hits. So as the number of required hits goes up, we require more peptides to have an effective response, but what you can see is that we can effectively reduce the uncovered fraction of the population effectively by adding a small number of 25 MER peptides. And then the same thing is true for the RBD domain, where we can have a rapid drop-off in the uncovered frequency of the population by adding a smaller number of 25-mer peptides. Now, the way that these can be delivered in the case of nucleic acid vaccines is we can directly incorporate a secretion signal sequence and an MHC class 1 trafficking signal into the peptide coding region, and each one of the bolded regions is one of the peptides, and we have linkers between them. And other studies have shown that one can directly express this construct, and the peptides are cleaved and presented on both MHC class 1 and class 2 molecules. So the way that one would actually, in the case of, for example, a messenger RNA vaccine, add this in is you could add in extra RNA molecules that encoded this particular sequence. You could actually control how much augmentation you want to provide, but depending on the ratio of the number of original S units that you put into the vaccine design versus the amount of augmentation peptides to actually improve the cellular immune response in response to vaccination. And so, in answer to the question, how can we improve COVID-19 subunit vaccines? The answer I have is that we can augment them with peptides drawn from other parts of the pathogen to improve the cellular immune response. And finally, how can we make COVID-19 peptide vaccines? It turns out that it's been shown that you can make peptide vaccines that don't include a subunit at all, and these peptide vaccines can activate the cellular immune system, which can prevent subsequent disease. For example, there's been a 2-peptide vaccine demonstrated against Ebola. So in order to do this, we used our same framework, but we didn't include a subunit as a starting peptide set. Instead, we did a de novo design, and we compared our de novo design using the combinatorial optimization procedure to 29 baseline designs that have been put in the literature in the past few months. And what we observe is that the top row here is MHC class 1 coverage, and the bottom row is MHC class 2 coverage. And what you could see, all the other designs suggesting peptide sets for vaccination for class 1 and class 2 coverage were able to surpass their performance typically with a smaller number of peptides, showing that combinatorial optimization is a great approach for designing peptides for vaccination for cellular immunity. And if we look at the trade-offs a vaccine designer has to make, we can choose different 25-mer peptide counts to achieve different numbers of peptide-HLA hits across a population. So with 24 peptides, we can achieve over 90 or 94% coverage with at least 10 peptide-HLA hits across the entire population. And if we look at this vaccine design, you can see that here is the coverage for various criteria of number of hits we would like to see against the HLA molecules. And on the right-hand side is the distribution of the number of hits in the population. So we can see the expected number is a red dotted line. And of course, this actually provides a fairly robust immune response compared to, for example, the RBD only subunit that at times didn't have any coverage at all of the population. So how can we make COVID-19 peptide vaccines? Well, we can do combinatorial optimization of candidates to achieve a desired population coverage. So just summing up, I wanted just to bring up how we're pushing forward on these designs. We're testing the peptides that we have predicted to be presented for their immune response, typically in blood from convalescent COVID-19 patients with 2 different collaborators, Steve Elledge at Harvard and Shizu Chen here at MIT. And to test our designs first in animal models, we're working with Jinsu Chen here at MIT once again, and also Chris Love is adapting his S-unit protein expression to include some augmentation domains. We're also talking to biotech collaborators about working with them on animal model augmentation of their vaccines. I think that many of the Warp Speed participants need to have a plan B in the event that the vaccines that they have developed don't provide a durable immune response. And finally, we're using this framework to predict individual display of peptides for the entire virus for risk prediction analysis. One hypothesis is that if your particular HLA type does not actually allow you to present a diverse set of viral peptides, you may be at greater risk for more severe COVID-19 disease. And so we're going to investigate that using data from the UK Biobank. So thank you very much. Once again, I'd like to thank all the people that contributed to this, Guy Liu, Brandon Carter, Trenton Bricklin, Sid Jane, Mary Carrington, Matthias Viard, and I'd be delighted to take any questions. Thank you very much.
Great, thanks. So as folks come up with questions, as a reminder, on the Participants tab on your Zoom window, there's a little icon that you can use to raise a hand, and I can get you to come off of mute. Maybe I'll start with one, David, while other folks are warming theirs up. I was curious about, with multiple peptides being displayed with the linkers, if there's any potential issues from association of those peptides to each other and correctly displaying so that all of them could generate a response. Is that something that is potentially an issue, or is that something that can be mitigated with the design?
One thing that could be an issue is that when you actually assemble things together, that at the junctions, there could be peptides that are created that you wouldn't expect. So we can scan for those. We also have looked at the relative disorder of that entire protein domain. You know, we want to minimize the disorder of it. And so that's another aspect of it. As far as we know, others have successfully assembled domains like that and expressed multiple peptides, so we're hopeful that it will function properly.
Great. Good. All right. We've got a question in the chat. Would a personalized medicine approach be possible where one delivered a vaccine with the best set of peptides for a given genotype?
It's possible. In fact, that's what a lot of cancer vaccines do that are therapeutic. Remember that one challenge is that a given HLA type for an individual will cause them to display a certain set of COVID-19 peptides. We can vaccinate for those to actually produce a better cellular immune response. What we try and do is build a universal vaccine that will cause everybody to display a large number of peptides. It is possible, however, to design customized approaches.
One follow-up I'd have on that question is this data that can be relatively easily collected after the new vaccines that are going on to market are emerging, so that we can understand which of those are having more potency within various groups to have perhaps greater predictive power? If there are multiple options, could somebody be typed and take a various vaccine that's out there?
Well, that's a great question. And one thing that we've been encouraging folks in the biotech field to do is to collect the HLA types of their clinical trial participants, so we can look at the relative efficacy of the vaccine in different HLA backgrounds. As far as we know, they are not doing that right now, unfortunately. One needs to get as you can see, that there's a fairly broad range of display of peptides across the population. And so we've identified particular HLA groups that we think are not going to present COVID-19 peptides well. They would need to be included in any kind of clinical study to make sure that the vaccine was effective in that population. One thing that perhaps is unfortunate is that the US government says that if a vaccine is 50% effective, then it can be marketed. That's sort of their bar. And it may be that if a large chunk of the population is left unprotected, that could create an issue later on. You saw the data earlier in my talk suggesting that older individuals which have a more senescent immune system, they actually may be harder to vaccinate effectively, and it may be they require more peptide display to actually get a good immune response in memory. So these are all issues that need to be considered.
Got a couple other questions coming in the chat. As a reminder to our participants and folks, it is easier if you are able to ask these directly, and I'm happy to bring you off of mute. Please let me know if you're having any challenges with that. And if anybody would like to ask their questions live, we're more than happy to do that at this point. Otherwise, we can start jumping in with some of these. David, can you see some of these from the chat window? And
I can. So, how are HLA peptide hits in an individual with a given genotype computed? Well, we first compute of all of the COVID-19 peptides, which ones are bound by each HLA allele. So we exhaustively look at all the possible HLA alleles, compute what's bound. And then for a given genotype, we can simply sum up the number of hits that we see in that particular genotype. So that was for Pradeep. And then Sebastian asks, what are some of the possible reasons that the display of a particular peptide on MHC does not necessarily correlate with how it triggers an immune response? Well, there's been some very interesting studies in mice where they asked what fraction of displayed peptides are immunogenic for CD8 or class 1 responses. And they found that almost all peptides are immunogenic in some individual. Some peptides are broadly immunogenic in many individuals. Here, individuals are mice. But this depends upon, the particular set of T cell receptors that an individual has, which is related to their genotype. So it's very difficult to be able to predict in advance which particular peptides are going to be immunogenic, save to say that current evidence suggests that the more diverse peptides you can display, the better off you're going to be, because it will increase the probability at least some set of them will be immunogenic. And Keaton asks, is it known how much of the COVID immune response is B cell versus T cell based? Is the optimal set of subunits the same for B and T cell response? So to get a good B cell response, you want to present an epitope that is going to be neutralized. You want the antibodies to bind to that epitope to neutralize the virus. The challenge I presented in my remarks was that those subunits, when processed and presented, may not provide a robust T cell response because they're necessarily subsetting the entire pathogen. And thus, it's not really known how important B cell versus T cell responses are. T cell responses are thought to be very important for durability of an immune response, but that has yet to be determined. One of the challenges of the current vaccine race is that we're really not going to know how durable the immune response is and how much boosting might be required downstream. Li Rang asks, will the augmentation construct be directly attached to the spike protein? And if so, how do you prevent it from becoming a distracting B cell epitope? Okay. So the answer is that in the work we're doing with Chris Love, yes, we're going to directly attach the augmentation construct as a separate domain on the spike protein, and yes, it could be a distracting B cell epitope. So that could lower the effectiveness of the spike protein itself. That's really not known. However, if we deliver it as a nucleic acid construct, it will not be attached to the spike protein. It will be a completely separate RNA molecule that will be separately translated, and it will not be covalently attached to the spike protein in any way. So I think that's it for the chat questions.
That is. We can give folks a few more minutes. What are tell us about what some of the next steps are for you in this process. And one thing that I actually have in mind in particular is there are some Europe opportunities coming up in which we might be able to get students available to collect data. So what ways might that be useful?
Well, this is a completely open source project, so all of our code and data are already pushed out to GitHub, and so people are certainly are capable of downloading our repo and actually playing with the code. I think that the next steps for what we're doing are working with others to, as I said, look at convalescent sera from COVID-19 patients to see which peptides are immunogenic. We really need to go into experimental studies to see how effective vaccines are. And I think a question that was posed earlier about the importance of B cell versus T cell response is really undetermined, right? There's another question here in the chat. Any thoughts on transpliced cellular viral messages and resulting epitopes and how that may affect immunogenicity? Not sure what a transpliced cellular viral message means in this case. So perhaps the person who, maybe Christopher can elaborate on that. Do you want to unmute Christopher and ask?
Yeah, let's see if that that works for Christopher here real quick.
Hello, I'm unmuted, I believe.
You are unmuted.
Thanks for taking the question. I came in late, so I may have missed the earlier stuff. I was thinking that there is precedence for viral proteins to be joined in a splicing mechanism to cellular proteins, and whether that might have any effect on the presentation of those peptides and resulting immunity, positive or negative.
Right, we have no evidence right now that SARS-CoV-2 peptides are being spliced together with cellular proteins, altering their presentation. That certainly would be an interesting fact if it was discovered, but right now we are assuming that that's not happening, and that the epitopes that are being presented by COVID-19 are native epitopes coming from its proteome. Now, of course, the proteome is mutating all the time, and we're trying to pick epitopes that actually are relatively invariant across these 4,000 different strains that have been sequenced across the world. That said, we'd be very interested in understanding if there are novel epitopes that arise through any mechanism.
Okay, so there's no evidence thus far for transplicing to occur.
Not to my knowledge.
Okay.
There's been a fair amount of RNA sequencing of the virus, and as far as I know, there's been no transplicing observed.
Okay, thank you.
One challenge we face, of course, is that none of the participants in Operation Warp Speed, or for that matter in any other commercial vaccine effort, have disclosed what's exactly in their vaccine. So it's not really possible to do a complete evaluation of all these vaccines because we don't know what's in them, which is a bit of a challenge. And I guess I personally would encourage people who are going to be developing these vaccines and getting literally billions of dollars from the federal government to do so, to fully disclose what's in their vaccines so that we can really carefully analyze them from an independent perspective.
David, is that just publicly disclosed and available information? I would assume as part of the regulatory process, they would need to be disclosing everything to FDA. Is that right?
That's not true.
Oh.
As far as I can tell, if you look at the clinical trial data, they tell you which general subunit they're using or what their delivery methodology is, but there's no sequence data available for any of the vaccine designs.
Interesting.
So you cannot get the exact sequences they're using, nor any of the targeting domains. For example, the Moderna design is a membrane-targeted spike protein, which means it's going to be stuck in the membrane. And if you look at the Moderna clinical trial results, what you see is there is basically no CD8 response at all. And so the surveillance function of cytotoxic T lymphocytes that are going to go around and provide sterilizing immunity may be absent in the Moderna vaccine, which could be a significant issue for the efficacy of that vaccine. Of course, it's early days, it's hard to tell. But personally, I think open sourcing all the designs for these vaccines, given the amount of money that's being spent on them, would be a great idea.
Yeah. Great. Let's see. Are there any other questions from the participants or the guests?
I guess I would just like to say, if anybody is interested in vaccine design, I would encourage them just to email me directly at gifford@mit.edu, and I'd be delighted to respond.
Excellent. Well, on behalf of everybody here today, I'd like to thank you for taking the time and telling us about the exciting work that you've been doing.
Justin, thanks so much for the invitation. Really appreciate it.
Thank you. And to all of our guests, thank you for attending today, and have an excellent afternoon. And we will see you again next week. Bye.